Right, good. Welcome, everybody. Panelists who wanted to be part of the audience, we'll finally get them to come up here. Could everybody settle? Thank you. The reason for this is really that last Friday we had a meeting, the Autism Tree Global Neuroscience Conference, at which these three people were speakers, and it's actually wonderful, and they're all very, very local as well. And I'll sort of vaguely repeat what I was saying earlier, which is that Alison and I have known each other since the early 2000s when we were in Rusty Gage's lab. Rusty is now president of the SOL. Liz did her PhD at UCSD and is now actually director of BI and director of the New Jersey Autism Center of Excellence. You went to Caltech after that? Yeah. And Leanne, although she started at other places, when I came to the SOL in early 2000s, you were there. You were doing so this is wonderful in the sense this is kind of my slow community all coming back, and their talks were so appealing that I asked if they would mind hanging on a week, hang on a few days, and they said that they would. I mean, Leanne's talk was autism, gaming, and attention, just so you know. Liz's was the earliest detection of neurodevelopmental derailment leading to autism is now within our grasp. It's a pretty intriguing statement. And Alison quietly said gene therapy and autism, although it's a lot more. Are you going to start? You're going to start? Yeah. So I'm trying. I won't do lots of talking because we know the time has been cut short, but a lot of Alison's work recently, a lot of the attention has been towards his work with organoids, brain cells in a dish, and there's just a massive interesting stories going back to, I suppose, 2019 was when you first did that really interesting paper. And then you were asked to comment on what it actually meant in terms of having mini brains grown in the lab. There are some of my colleagues who say, no, these things will never be conscious. Lead researcher Alison Moultrie said. Now I'm not so sure. So a huge amount of stuff going on in terms of organoids. And then there's a lot of stuff going on as well in terms of gaming. There's a lot of the things that are kind of negative in terms of that. And Leanne has some wonderful positive things to say about all of these things. What intrigued me a lot was the overlap and how to explore the overlap between what you guys all do and autism in particular. And I mean, one example would be that one of your papers, one of the posters today, transparent graphene electrodes and 2-photon imaging revealed functional connectivity between cortical organoids and mouse visual cortex. Madison Wilson's paper, but it also has on it Alison Moultrie, Fred Gage, Anna DeVore, and that poster, I am just told, was actually accepted for publication today.
Yeah, that's what I hear.
So, I mean, everything is now. And you got any more to say about that paper? And then just roll.
No, I better get started and then we can discuss that. So, yeah, for those who are not familiar with that, this is a bottoms-up study of the brain. So we don't really study the brain. We try to recreate the brain. And by doing that, try to understand the fundamental mechanisms of the nervous system. And for autism is something quite important because autism is, has a strong genetic factor. So it actually starts in uterus. So you do not acquire autism through your life, you're born with autism. So we have a way to create stem cells from people with autism so we can capture their genome in stem cells. These are pluripotent stem cells that basically can make any other tissue from the body. And so, yeah, the reason why we're doing that is just to take out the early stages of brain development from the uterus, from the womb, and take it to the lab. So I can study how the brain is formed in a very controlled situation. So I don't have to rely on non-invasive technologies such as ultrasound or things like that. So the way we do that is to take the stem cells, and then we have, that's what my lab does, we create recipes for the different parts of the brain, or for putting parts together. So this is a recipe for the cortex. We start with single cells. These are pluripotent stem cells, as I mentioned. We can add factors to the media of these cells. So they neuralize and they acquire the cortical fate, and then you stimulate the progenitor cells to grow. And we remove the growth factors and we let the neurons to form networks. If you've never seen one of these before, this is how they look like. This is a postdoc in the lab, and she's handling a plate with about 3,000 of these organoids in these wells. So each one of these white dots is one brain organoid that contains about 5 million neurons, same as a bee brain. And they will grow up to 0.5 centimeters, so you can actually see them. So not only they grow that, but they acquire some very interesting anatomy that resemble the human fetal brain. There's a ventricular zone where the progenitor cells are. They migrate from inside out, so they form the cortical plate. And so we can study all these processes in the lab. Some people ask me what kind of cells you have in there, so we can destroy or explode these organoids and analyze each one of these cells using a technology called single-cell RNA-seq that gives you the identity of each cell. So you can see that at 4 months of age, we have a population of glutamatergic neurons. That's how the brain does. It forms all the excitation first, but that population is already forming a discrete population of GABAergic neurons that would become about 20% of the entire population later on. There's also the production of glia cells, mostly radioglia, that helps the migration of the cells, as well as astrocytes that will peak when they are about 6 months of age and will help with the formation of the synapses in these very early stages. So we also look for the formation of these networks over time. Remember, a brain gestation takes about 9 months, so we can keep these organoids alive for several years. And by plating them on top of the multi-electrode array, we can capture the activity of each electrode over time, creating these very dynamic raster plots. So what happens is it's very interesting from the electrical activity. We start with random noise in these networks, but they suddenly arrange and start to see synchronized bursts. This synchronization will get way more frequent over time. By the time that they are 8-9 months, that's when the inhibitory neurons start to function, and you start to get a nested oscillation in these networks. So these actually resemble something that you guys are used to, which is several frequencies that you can capture through EEG. So we actually analyze if we do see those brain oscillations. That was a collaboration with Brad Wojtek here at UCSD. The oscillations start to emerge when the organoids are about 4 months. By 6 months, you have this stereotype type of oscillations. By 8 months, they become highly complex, and the complexity just increase after that. But most importantly, we compare the merging and the dynamic of these complex oscillations with several preterm human baby's brain. We capture the EEGs and we create a simple regression model. And what basically this data is showing is that compared to the human data in black, the organoid data in blue actually forms the same trajectory, meaning that it's mimicking what the brain is doing at very early stages, which is critical for someone studying autism, because that's where we start to see the problems with the networks. So we are my lab and others are improving these methods by adding input and output, meaning sensory information. So these brain organoids can start interacting with external cues. This is one example. For example, we can create a retina in a dish. We can create a thalamus in a dish, and you can create the cortex. And then we have reporters, for example, green here markers the photoreceptors, and the axonal projections is in red. So you can put the pieces together and recreate this visual circuitry. And the hope here is to stimulate the photoreceptors while recording from the cortex, so we can create a visual cortex. So that's one idea. The other idea, it is to give like an entire body to these organoids so they can start interacting with the environment. This is a collaboration that I have here with Seni, trying to understand how these networks will emerge. So we work with a brain-machine interface. So what you're seeing here is a robot that will start to learn by a closed feedback loop from an organoid that is sitting inside one of our incubators. So we are populating this robot with several sensory information. For example, the infrared here that will tell or will stimulate the organoid as approaching a wall, so it will pause and change directions. So by doing that, we hope to achieve two things. One is to further mature these cortical organoids so it can reach other levels of complexity that we currently don't have. The other thing, it's more applied, is to learn how the human brain learns, so we can redesign AI algorithms that will be more organic inspired, more energy efficiency, things like that. So overall, I mean, I hope I convince you guys that we have a stem cell model now that mimics the early stages of neurodevelopment, and we are doing that for several types of autism. We are starting with the ones that are genetically defined, that you can actually sequence their genome and find the genetic alterations. Here is just a few of them. Some are monogenetic, some are polygenetics. We created these organoids, we look for deficiencies in each one of them, and then we take advantage of the changes compared to control organoids to test potential therapies. For example, we can do drug testing to see if we can rescue some of these phenotypes. We can try for a gene therapy or a genome editing, trying to fix the mutation directly into the DNA. And we are hoping that by doing that, we can accelerate clinical trials. Most of these models, they don't actually have an animal model that can be studied. So these families are with no hope. So we are hoping that now with a brain organoid, we can actually speed it up the whole process to go into clinical trial. So that's my short message here, just thanking the families that participate in this work and members of my lab. Thank you.
Have any questions right now? We'll do all three of them and then have a conversation at the end. Do you have any questions specifically about Allison's tool? You do?
Very exciting. So you show these robots that are roving around based on the so what is the magic behind what is happening? What are the organoids doing?
So the organoids are doing nothing. They are sitting inside the incubator with the multi-electrodes surrounding them. And we're just capturing the activity. It goes to a computer, the decoder, that will tell the robot what to do. And as the robot starts to stimulate the organoids, I mean, we see the rearrangement of these networks that now give second command to the robot. The goal is, you probably heard, like I think 2 or 3 weeks ago, someone published some paper showing that a network of neurons can play Pong and can learn faster than AI. So if a network of neurons can do that, you can imagine what an organoid can do it. So the computational power that you have in something like that is really something that we haven't explored yet. So that's our initial idea to get in that interface. Yeah.
Do you find differences in the learning patterns of the organoids that have autism-related genetics versus neurotypical ones?
Yeah, we haven't done yet the learning paradigm for that, but based on all the alterations, both on the structure as well as synaptogenesis and network formation, the answer is probably yes, but we haven't tested yet. No.
Anyone? Somebody with a hand up somewhere?
Yeah, thank you. I guess we were talking over here about, I'm sure you get asked this question all the time, but the ethics of these. At what point, I mean, if it's very able to learn and it's controlling a robot, what's the line of, like, how do you think about these ethically?
Yeah, so that was a great question, and that's exactly the reason why I start having a dialogue with philosophers of the mind as well as ethicists, people like that. To my surprise, I mean, they don't know, because this was not in the philosopher's book. I mean, that was something that we just recreate now, and we have to learn how this will evolve. The most pressing issue is if they start to acquire self-aware or some level of consciousness, and that might put us on a gray zone, and we might have to decide what is the moral state of those entities. Should we treat them as humans? Should we treat them as animals in the lab? I mean, what is exactly what that we're doing here? So we don't know. Nobody knows. So we're observing how this field is evolving.
Yeah. So why don't we just move on, and we can add some more stuff at the end of this. Do you want to speak? Thank you. This town, by the way, the County of San Diego, is the first public neurodiversity workforce initiative of its kind in the nation. That was done by the Autism Free Project Foundation, working with Supervisor Tara Lawson-Raymer. That was passed in June, so unanimously 5-0. So it struck me as great news that this should be the first place that would actually be encouraging autism and ASD's workforce. And then it struck me that, what? This is the first time in the first place? And I mean, this seems to be way behind on all this. So I hope things speed up a little bit.
Okay, so
Yes, ma'am.
can you hear
Off
me?
you go.
Yeah. Yeah, it's not on. Whoa. On there. Hello? Yes. I can talk very loud.
There you go.
Okay, good. All right, so I'll talk about the earliest detection we have on autism. This is a large project, and I'll be talking primarily about work I did with the top row and my students here. But I also did some work during my sabbatical with primarily Simon and Rusty and Terry. But I won't have time for that to cover that. So autism has increased exponentially in the past decade or so. And there is a kind of an urgency to understand these earlier, because the average age of diagnosis is about 4.5 years of age when the circuitry has already been formed, and a lot of the plasticity that we hope for is kind of past. Nonetheless, babies are born with several structures that are already working, and more importantly are quantifiable. And one of them is the brainstem, which is the conduit between the cerebellum, the spinal cord, the neocortex, and subcortical regions, and it's measurable very early on. I want to just emphasize the importance of this first year of age in a human, which the humans stand over here in this power law that relates the brain weight with the the time that it takes to actually walk. That we're altricial mammals rather than precocious mammals, like the horses that are born and immediately trot and run. And so that first year of age is really important, and obviously for the plasticity. And there is a method that it's already a routine auditory screening test called the Evoked Brain Stem Response, or ABR. And it's done with portable systems, it's done routinely. And what we didn't know about it is that there is a lot of information, the waveforms that the system that does it output. There are well-established latencies of these 7 peaks that have been characterized and correspond to these regions of from all the way from the auditory nerve, the pons, the midbrain, all the way to the primary auditory cortex. And in the particular data set that I'll talk about, we had access to different decibel levels all the way to audible levels that we could analyze. But one thing I noticed was that the response waveform that we get has an underutilized information in these wiggles that precede the spike, and also the wiggles that after the spike. And so if we zoom in, you see that there are a number of peaks there that can be analyzed. And also, if I zoom in into the spike, there are a number of features of the waveform that can be utilized. And so one problem that we have in neonatal stages is that everyday counts, they grow very fast. And so we would have to account for anatomical differences. And this is currently not being done by any of the methods of motion analysis or brain waveforms. And so we just collected a number of babies that went on to develop typically, and babies that went on to receive a diagnosis of autism. So we could go back in time and look at their waveforms. And indeed, their rate of growth is nonlinear. And so we needed to account for these allometric effects affecting the wave. So what we did in this case was normalize all these wiggles and the peak, and came up with a new data type called micro movement spikes. And these are spikes. So these are in the range from 0 to 1, and they come from the fluctuations away from the empirically estimated mean of the original spikes. And so they're a scale out for allometric effects. And so this is in contrast to what the field has been doing all along, which is essentially an example of a positive rectified waveform here. They essentially superimpose it and then assume a Gaussian distribution and a Gaussian mean and 2 standard deviations from the mean, and everything else above that is treated as gross data. So it's really wasted. So what we're doing is applying our micro movement spikes, reformulation of the fluctuations in the data, and empirically estimating using maximum likelihood estimation the family of distributions that best characterize each one of these processes. So in this case, we found that the continuous gamma family of probability distributions very well fits very well the biorhythmic data, not only from this type of data, but kinematics, any biosensors that essentially you can analyze, and we can track the changes in distributions as they transition from basically a spontaneous random noise to well-structured noise to predictive signals. And this is general for all types of data we've collected, but in particular for the neonatal data that we've collected over the span of 30 weeks with an app at home that the parents can do. So we have a parameter space that is interpretable, and over here are essentially distributions that are in the exponential randomless, memoryless random distributions. Over here are distributions that are heavy tail, and then over here are Gaussian-like distributions. And so we can track any process over time like that, longitudinally speaking. And we did this in a cross-sectional data analysis of autistics, and found that typically there is a maturation power law characterizing their kinematics for purpose of movements. And that is progression is absent in autism. And that gave us a sense of the need to understand these fluctuations across other data types. So these are essentially defining a concrete target for treatment. And we can then distinguish unambiguously autism from non-autism using these methods. In the case of the ABR waveform, these are, for example, the prominences. We can not only distinguish the different levels of decibel of the sound that we administer to babies, but also whether they were preterm or full term, or whether they had a diagnosis of autism, or they were typically. Yes.
I'm sorry, I missed it. What are the horizontal and vertical axes?
Yeah, so this is the shape of the log. There's a log plane, and this is the shape, the gamma shape that we empirically estimate from the distribution from the proms that we keep. So we keep the timestamps, and those are the prominences, the amplitude in microvolts, but normalized to these micro movements waveform. And then this is the scale, which is the dispersion of the distribution. It also happens to be, in the case of the gamma family, the noise to signal ratio. So the variance divided by the mean. So that, yeah. So, I mean, skip over that. Just I want to get to the latencies, because I think this is more relevant to people that are working on speech and spoken language. But essentially, the blue is the non-autistic neonates, and those are unambiguously delayed with respect to the neurotypicals, but also, so you can see them. These are the actual latencies, and this is the empirical PDF, which I will examine in the next slide. But each one of these peaks from 1 to 7 are separated, and they live in a different, they are characterized by a power law that lives in a different intercept. So this is now zooming in those distributions, and for each one of the peaks, you see not only that the autistics are delayed, but also that they have a very narrow bandwidth of information, as opposed to the neurotypicals in blue here, which have a broader range of latency, so they have access to much more information. This is the cumulative delays, and you have 1.74 milliseconds in latencies, and this is in a system that computes on microseconds time scale. So this is a very delayed signal, and finally, sort of to emphasize the importance of the brainstem, and perhaps this carries information with explanatory power of all these behaviors that we see in autism, spinning, shaking, and what are called obsessive behaviors. I'm going to skip over that, but essentially we reproduce that from 1.8 to 6.8 years of age. And finally, turns out that the entropy of these distributions also separates preterm babies from full-term babies. And we had a chance to look at a large number, 233,000 plus babies. And over the course of 8 weeks, we could see that the preemies were not changing the latencies, whereas the full terms were actually decreasing their latencies of these waveform. So in conclusion, we have information buried in the gross data that we tend to discard when we take grand averages of the data. There is broad variability in the latency ranges, and these are quantifiable delays. And with very little cost and very little effort, we could actually develop a screener for prodrome autism using a test that is already in use, at least in the US, at birth. So that's the conclusion.
I do have it.
Um,
So I'm fascinating. Thank you very much.
Yeah.
I'm trying to connect between ABR, which is used prelingual
Yeah.
as a test to see if things are getting clear or not. But my little knowledge of autism with respect to language comprehension, once they have acquired language, is there something there similar to what you were showing in ABR? I'm trying to connect the prelingual and once they have acquired language.
So yes, so there is, so autism is a very broad spectrum. So 30% go on to not acquire spoken language. So they're non-speaking autistics. And we know very little about that population because it really hasn't been studied. But for the other rest of the autistic population that has been somewhat studied, I had to say the current inventories, that clinical diagnoses are flawed, statistically flawed. And so part of the consequence of that is that we get 4 to 5 boys for each girl. So we don't know much about the girls either. So any given random draw of the population is not enough statistical power of the study in question to know about the girl. But now let's, like, what we know about autism really is upper and middle class white America and the so-called high functioning, which is a level that gets to acquire some language. And the reason, a lot of the reasons behind their awkwardness or the language acquisition problems lie precisely, and we're proposing, it's an invitation to think about it, on these delays, because conversation is very dynamic. And if you have 1.72 millisecond delay that remains throughout your life until you are diagnosed with autism at 4.5 years of age, you've been living your entire life in a different perceptual bubble. I mean, you have developed a completely different way to integrate other sensory signals that have disparate latencies. And so you have information traveling on the order of 100 milliseconds, information on the other microseconds, information on the other seconds, and you have people around you that are also processing information with those different latencies. If you have this delay, so language acquisition, social interactions, and so forth is inevitably going to be impeded because of all those differences in latencies, and not to talk about inertial times, delayed times of movement itself, right? And transduction and transmission time of the motor signals. So sensory motor integration takes time. And as we saw in the last talk by Gopal, I think that you can see that if you have already a system that is not processing at the latencies of the regular system, it's going to be very difficult to establish communication. Just even from the standpoint of information processing. Yeah, so yeah, it does impact.
Right, thanks very much. Let's move on to Leanne, and thank you very much. the work she does at Northeastern, Leanne was also a co-founder of Brain Leap Technologies, which was founded to make attention training games, which developed at UC San Diego. And I sent her recently, because it struck me that games were one of the things that you could have some very negative side effects from. So I was wondering what her answers to this were. I was just playing devil's advocate.
So I get to give my talk first?
I sent her a couple of papers. One was called Upgrading the Protection of Children from Manipulative and Addictive Strategies in Online Games. And the other was called The Playing Brain, the impact of video games on cognition and behavior in pediatric age at the time of lockdown, a systematic review. And then in Cell Press in Neuron, dated December 7, 2022, it's now the future. Paper is titled In Vitro Neurons Learn and Exhibit Sentience when Embodied in a Simulated Game World, and they put it in a kind of a Pong situation. So this stuff is really, really hot. Off you go.
Well, thank you, Roger, with that introduction. He knows I like a challenge, I guess. Thanks. So let's start with autism, attention, and gaming. And so I was here for many years, and most of the work that I'm going to talk about today was actually done here at UC San Diego. But I really want to orient you first to the importance of play for the development of skills. Animals across the kingdom play, right? Humans play. We actually should be playing more in order to learn things, to learn our skills, even as adults, right? So we'll get back to the bad parts about play, maybe, but by and large, play, whether it's video games or not, is a good thing for us to simulate interactions with each other, with virtual creatures like these young girls are doing. So why am I pitching video games as a way to examine behavior, a way to assess, and a way to potentially intervene in some aspects of, as I will get to, attention? And it's because video games and games that are not video based actually can really support aspects of autonomy, giving some agency or purpose to the players. They encourage mastery. They have aspects of uncertainty to them that allow people to engage, like models of explore, exploit behavior. They're very modelable. In fact, there's been a really excellent write-up about how AI is arguably has advanced as it has because of what you can do with AI in games, right? So it's immersive, competitive when it wants to be, cooperative when it wants to be. You can engage with your peers, and most importantly, engage. So these are the kinds of things I want to use when I'm developing an assessment. I want to put people in the best place I could possibly put them to measure things, including kids with developmental differences. So about 10 years ago now, video games for cognitive training sort of came into view. And the paper that I think kicked that off was from Sean Green and Daphne Balvier that was published in Current Biology, showing that there are links between a genre of video games that is known as action video games and some skills in the learning and perceptual realm, particularly improvements in aspects of perception, visual spatial attention, movement skill, and working memory. So we asked if we could maybe use video games to try to train aspects of these skills in kids for which they were weak. And in particular, we were interested in designing a suite of video games in a very particular way, not your standard video games that you played with a mouse or played with a touch screen, but video games that you played with your eyes. And the reason for that is because your eyes are really tightly controlled to how we shift our attention. We move our eyes about 3 to 4 times every second. And so if I'm really interested in training aspects of attention and the orient aspects of attention, using gaze is a really great harness to get in there. We also really wanted to make these easy to access. We wanted them to be available to people either at home or at school. And we particularly wanted to target a wide age range and starting with young children. And we found in particular that kids who sort of were past the age of early intervention were really interested in doing this because there wasn't much for them for training. So what specifically were we targeting? This is my colleague, Jean Townsend, who I had the pleasure of working with for over 10 years here at UC San Diego. And she is looking, she has done studies on attention orienting. Attention is a very broad construct, but attention orienting is this notion of sending, if you're looking at a particular thing, sending your mind's eye someplace else before you look, or without even making that look. And so in this study I'm showing you right here, this is something that we would call a Posner-like cueing task. So you are given a cue, and then a target appears, and the cue is spatial. It's either going to happen on the right or the left. And that the time between those cues can be either fast, 100 milliseconds, or slow, 800 milliseconds. 100 milliseconds is very fast, even with respect to making an eye movement. We tend to make 3 to 4 a second, whereas 800 milliseconds is quite slow. Regardless of whether you make this cue to target interval slow or fast, individuals who are not autistic do very well. Their performance is quite high. But kids on the autism spectrum, much like you just heard from Liz, have problems with temporal dynamics. The fast changes in the environment, even in this visual perceptual task, end up being more impacted when the information is presented quickly, fast, with respect to the target that's about to appear. So we wanted to take advantage of this voluntary saccade circuit, right, which is throughout the brain. And we can easily, as I was saying, think of it as the voluntary attention circuit and use gaze as a harness to try to change this behavior. So I was just showing you aspects of attention that's different in autism, and now I'm showing you saccade control. That is the overt manifestation of attention. And here I'm showing you data on just a center out saccade task. I'm asking people to look at the center of the screen, and then one big target appears in the screen, a little Cheerio, and all you have to do is align your gaze to it. And the autistic participants take more saccades to get on target. It takes them longer to launch more saccades to get on target, longer to launch their first saccade, and analogous to this, their accuracy of their first aligning saccade takes longer. So again, the way of addressing this was by making a suite of video games that you play with your eyes. This simple eye tracker, which is commercially available and fairly inexpensive, can be added to a laptop or a desktop machine. And we've made video games in the Unity game engine with a series of training principles. I'm not going to go through the video, but if you're interested, send me an email. I'm happy to send you links to it. But we've made different video games with different kinds of training principles that we can help kids make those fast shifts of attention, also steadily fixate on an item so that they are ready to make a fast movement when they need to, and also to suppress movements to things that are not informative in the scene, right? So you're not supposed to just be going to everything, right? There are some things that we're supposed to not look at right now. And that is an aspect of training in some of these games too. Of course, the kids get better at the games, right? If they wouldn't be very interesting if they don't. But we have a suite of tasks that we measured behavior in before and after, and we see improvement in the kids in this very small clinical trial, pre to post. Now, again, these kids play the games at home. They played for 12 weeks. We didn't have to do a lot of monitoring while they're playing. We just brought them back in because the eye tracker sort of enforces the fact that you're either playing or you're not. So they got better at steady fixation. They were better able to inhibit a movement in this task that some of you may be familiar with called an anti-saccade task. They were able to inhibit a movement to a target that appeared and instead make a movement to the opposite side. Got better at that with training. They were in a divided attention task, but they were using a simulated driving situation. They were able to divide their attention more accurately. We did not actually see any significant improvements in road excursions. I'm going to blame the 9-year-olds for that because they really like to crash the simulated driving, but that's okay. We really need to get better. Much like I said, that my driving vision now is to think about not just interventions, but assessments that we can do, because it's hard to measure these things if you don't have good assessments. And so we also made a suite of assessment tasks that can help us, that are separate from the games, but can actually also be done with an eye tracker, also remotely. And this guy up here is actually, that is the anti-saccade task. You have to look at this fisherman, and he's got a buoy, and then either a fish pops up that you look at, or a turtle pops up on the other side, and you need to look at the opposite side. These tasks, like any game, good game, generate data. This particular task is called Soups On. Here it is down here. Your job is to look at the fire to keep it going. And looking at the fire to keep it going means that you're not getting distracted by all the things in the background. All those things meaning out here, there are little animals that come up, there are eyes that pop out of the bush, and there are all these things that are keeping you from your task and keeping your gaze right there. This is a very sensitive task that compares well with a standardized attention task. It does a great job. We've done some of this. We published a small conference paper showing that over time, kids who play the training games also perform better on these assessments. So this is something we can do outside the lab. We've also partnered with a school, a lab school in Florida to do this, and saw that kids improved in their fast gaze shifts, inhibitory control, and fast attention shifts. This was done in partnership with that company that Roger told you, Brainleap. Across multiple kids, we saw in these games that are about fast attention shifts and inhibitory control, we see improvements. So I'm gonna just take the statement that not all games are bad. There are some games that can be quite good for you depending on how you make them. We have taken this a step further and engaged people in the community. As Roger said, there are unfortunately few opportunities for autistic young adults to get jobs. So over the past, I guess the first one was here at UC San Diego in 2018. The past 5 years, we have been engaging youth in internship programs so that we can help them upskill to get jobs, because the folks I was working with in the at-home clinical trial with a very early, often breaking eye tracker, I got to go to their homes a lot. I got to meet them, get to know them, get to know their families, and I learned that they were excited about this clinical trial because of the games, and they were writing their own game narratives, and they were making their own art, and they were even writing their own games in code, and it was locked in their personal space. It wasn't necessarily getting into the community. And so this is something that the San Diego Foundation initially funded, and then we went on to get NSF funding, and we're going into our 4th year of the Future of Work program, and it's been fantastic. And so this is something that we're definitely looking to take further. You will recall that I was introduced by Roger with a bit of a challenge, that games are not always good. So there are certainly beneficial effects of games. I remain, and I've gone actually to Northeastern largely because I get to be dual appointed in a program where I can explore my cognitive neuroscience side, but also collaborate with people who focus on game design so we can make better games together. We can really try to understand the benefits of play, but there are challenges. I would not tell you, I've got 2 children personally, that all games are fine and good. There are games that are specifically designed to manipulate. We live amongst the attention economy. We're immersed in it. It's not just games, folks, but games can be really good at trying to get your eyes back in the game again and again and again. And our youth, and some youth more than others, are not mature enough, they're not ready in order to engage that and know how to say no. So and we should discuss potential solutions to this problem, but I really think that it's beyond time that we should have content. It's not, ratings is not even it, but if anyone's seen Common Sense Media, I was a big fan, particularly when my kids were younger. I think that this is the same thing along different dimensions. I'm not even talking about violence, although of course that should be there, violence in games, but games that actually have those principles that are addicting. It's easy to see it. It's very quantifiable if it's there, but we should know it. Parents should know it. You should know what's going on, what these kids are playing, and be able to say no if it's not for you. And I wanted to just take a moment, since I have your attention here, and tell you about a really cool opportunity that's coming up. The IEEE conference on games is going to be at Northeastern University this August. And I'm super excited to be one of the chairs for this conference. The papers, which of course you're all going to submit to, right, are due March 17th. So I really hope that those of you who are interested in games for research purposes or otherwise consider coming. And then back to you, Roger. I know I didn't do your ethics thing the way you like.
No, that's fine. I was just, this makes the point. I was curious to know in discussion with Gert how much autism should be intersecting with research programs of folks like you who work IEEE areas, and that obviously is one of those. I had a question, which was the ones when you were doing in South Florida and so on. How much were the games that you designed specifically with people on the spectrum in mind useful in helping you to understand games for people who are not on the spectrum?
So thanks for that. So I've learned a ton even in the year and a half I've been at Northeastern, in part because most of the serious games that are out there, games for training, games for education, are lousy. I mean, we can do a much better job really in engaging kids. So what I've learned is that most of the kids out there who put these games in front of them, if they are savvy consumers and they're playing Roblox and they play Minecraft or what have you, then they'll play this in school. But when they get home, good luck. because you're now competing with games that had millions of dollars of investment into design. And that's not where we are. So I really think, better design.
Yeah, this is Neil Postman's classic amusing ourselves to death, isn't it? Were there any you want to close down fairly short? Are there any questions? No? Let me just go there.
There are some questions on chat. Maybe in the last 10 minutes.
Sorry, can you hear me okay?
They can hear you.
Oh, okay. I was just curious with the small clinical trials where you saw those improvements, if parents reported seeing anything transfer outside the study in terms of their functioning.
Yeah, thanks for that question. We had multiples of those, and unfortunately, it doesn't count. And I mean, like, so this is part of the chicken and egg problem right now, right? Everyone's looking for transfer, and the grumpier among our colleague set said, there's never transfer in these games, and I say, show me the mechanism, the tool you have for measuring excellent transfer in real world things. So yeah, we see that in parents. It's just that it's hard to know. They invested time in helping their kids play, right? Like there's a bit of a demand characteristic. Through Brainleaf, we've seen kids that did amazing things. Was that us? Yeah, I hope so, but I can't say for sure. So part of it is that I really think we need better tools for assessing things in the real world.
If you're
Thank you.
training with, sorry.
No, just thank you.
I want to follow up on that. If you're, by the way, I'm a big fan of games. I've like to use games in my research, and I'm like a natural language guy. I'm a big fan of naturalistic tasks and stuff. And I think neuroscience and psychology research using video games is the future. So I want to preface that. I like the trajectory, but I'm also one of these people that's skeptical about the generalization of these games because I don't think it's really been shown very well. The classic psychology attention task is very similar to the task you're building into the game. It's just that the game task is interesting and the psychology task is boring, right? So the fact that those generalize is kind of not that surprising. It would be more convincing if like their grades improve. You're increasing attention, right? That's where you're hoping you're increasing attention. One of the big problems kids have in school is it's hard to pay attention. So you think the grades would increase after this intervention? Is there any data like that?
So this is exactly why we wanted to be in schools. The proximal thing that I first want to try to address is reading scores. Time on math or reading scores. Those are the really like basic things that I think that we can move the needle on, and we need to be in schools and have that kind of trial. Like so that was an after school program. That's definitely where we're going. But yes, absolutely. And you're not wrong. Like we shouldn't be measuring these same things. It's just Kurt knows and some other folks know that we've been trying to measure behavior in a real world interaction. That's hard. I can't necessarily do that in many people, but I think that is actually one way also to get there. It's not going to scale well in schools, but this other way. Yes, absolutely right. And I think the skepticism is more.
Want to bring up questions from the chat or should I ask?
You said there were questions on the chat.
Yeah. There are some chat questions, yes. And perhaps, Marty, you raise your hand. You want to unmute yourself and ask questions? Over Zoom?
Maybe you can't find.
Go ahead.
So, I don't know how to exactly phrase this question. You were talking about the speech input and how normal and ASD people, and Leanne's work has been mostly on visual cues, it's a card and how they control it. But there seems to be something more related to coordination of multi-sensory integration and motor coordination. Now it looks like both of you guys know about this, but you didn't talk about it. And that's the part that I'm most interested in. Multi-sensory fusion and the motor control that comes. I mean, that's how we learn to walk, right? So
So I'm just gonna say something brief, and I'm glad you're really excited about it, and I am too. We're having a really hard time convincing the autism community that temporal dynamics are a problem in autism early on. Like you early on, later, whatever. Like, and so, and I'm sticking in a single dimension like saccades. Moving to multisensory, like good luck with the NIH, right? So it's, I don't, it's not really you're wrong, it's just that for the way in which we investigate things, like kind of have to push on it in one area, I think, at a time. But I like the fact that we're both seeing something that has suggested there's little challenges.
Yeah, so the motor control space, it's actually random, depending on what apply or deal with it. We made a discovery well over a decade ago that there is excessive motor noise across all the bioregions of the nervous system, of the autistic system. And we even broke it down into the autonomic nervous system, with heart, peripheral, with limbs, and involuntary movement, so for involuntary tics and undesirable noise and randomness. We discovered that our law that explains human maturation and not provided evidence for how to actually download that noise and bring the predictive code into play and so forth. And I have to tell you, I have survived because I'm very creative at creating revenue streams, and I have raised $8.5 million from my research on my own. I became an expert on this and how to create technology that raises funding. NIH is not funding this research. NSF is not funding it either because we need to the understanding of the most complex problem the brain has to solve, which is to develop motor control over central power generators, reflexes, and autonomous systems that we are born with already working, and then extend that to a social interaction system. Another human that has also 600 plus degrees of freedom that brain has to control, and our brain, and so on. It's a very complex problem. Been working on it for 20 years now. And I tell you that the NIH and the NSF need to get a clue. Whoever is running that show, they need to get a clue because they haven't got this. AI is working with a platform that is primarily based on vision and speech. You have not seen yet an autonomous robot out there, and you won't see it for a long time because the fundamental principles of autonomy, of agency, which is the balance between autonomy and control, are not there. right? Studying therefore these models, I mean, I have a background in mathematics, I'm talking here. I know what I'm talking about. And I've been dismissed. For 2 decades, I've survived it because I know how to do it. But and Leanne, the same, but we share the same experience.
We need more engineers.
Yeah.
And, no joke, no, seriously, because the fact that the psychologists, the clinical psychologists, I mean, I've had grant reviews that said there are no motor problems in autism.
Yeah.
It's not part of the diagnosis.
It's not
Like,
part.
well, look at that. Ask any parent if there are motor challenges in autism, and you'll hear a very different story. But it's a bit of a problem in terms of breaking in and like really trying to doggedly show documented data. And when you don't want to see something, it's really hard to see it.
The ones that got it now finally are the insurance companies, because none of the therapies for autism work. And insurance companies are losing tons of money. And on top of that, it's a multi-billion-dollar industry. So the therapies that are by mandate already imposed on taxpayers in the US are now creating so much revenue that private equity firms are dismantling and buying them. So most of the time I get contracted by therapies and big companies that are nationwide, so that we provide our biometrics for them to show progress to insurance companies so that they can get reimbursed. And that's where this is going. That's what we should talk, and this is where we should strategize, because it's not the NIH and the NSF that are going to fund it. No.
Yeah, a lot of politics involved. We're at the end. I wanted to ask you one more question, see if you can put a Cheerios on this before we close. Just on this bit, the stem cells, I mean, that legislation, CRISPR, Jennifer Doudna, making up various arguments and so on and so forth. Now organoids, where is it at in terms of your to be put in handcuffs?
So, I mean, and you know really well, Roger, the stem cell is always surrounded by controversies, right? I mean, we go back to Dolly the clone that surprised everyone, that scientists can clone an animal, but the research has been going on for several years until we reach that point. And that's the mistake that I don't want to make with the organoids. That's why once we start the dialogue soon, well, we already start talking. I mean, it's already in the news, kind of the mainstream media now. It's much better than Dolly the clone. And my feeling is that as any other technology, as soon as you start to see the good, as soon as the technology proves itself, it becomes acceptable. I mean, blood transfusion was controversial, organ transplantation was controversial, and we passed through that. And now we take it, we accept it, and there is no problem. I think with all the ethical potential controversies here with the organoid, at the end of the day, we are finding new treatments, we're helping people, and I think society will embrace that.
Yeah, so that again, the engineers need to talk to ethicists as well.
Yes.
Which is what we'll be doing tomorrow. There'll be a talk from Laura Cabrera at 2?
Yes. 2 o'clock.
2 o'clock. All right. Were there some other questions on that, good?
We have some, I think we covered.
Are there any
I can
more
read some
questions?
questions if you like. Yeah, but
At the end of it. All right, so let's leave it at that and thank the panel. Talking about, I mean, it's just immensely important. I'm just ready to see more, even more. Well, so that'll be great. Thank you.